Okay, let's unpack this. Imagine having a true shortcut, not just to, raw information, but to deep, nuanced understanding tailor-made just for you. Today, we're diving into something genuinely revolutionary in the world of biology, CellFM.
Yeah, it's pretty exciting stuff.
So our mission today is really to explore this fascinating breakthrough in understanding human cells. But maybe first, let's set the stage. What's the core challenge we're tackling here?
Right, so you have single-cell RNA sequencing,
Yeah.
scRNA-seq for short. It's completely changed molecular biology.
Right, let's just see what individual cells are up to, gene by gene. Incredible detail.
Exactly. Amazing for seeing the diversity within, say, a tumor or an organ. But, and it's a big but, you get this absolute data tsunami.
A tsunami.
Yeah, and it's messy data, it's noisy, it's sparse, meaning lots of missing values, and it's riddled with what we call batch effects.
Ah, batch effects. That's like variations from different experiments, different labs, different times, almost like static on the line.
Precisely. Think of it like trying to piece together a huge conversation recorded by 1,000 different people using different phones in noisy rooms. The core message is there, but it's buried.
And the existing tools, they struggle with that.
They often do, yeah. They can get overwhelmed by the sheer scale and complexity, or they can't properly correct for those batch effects, so you might miss the real biological signal.
Okay, so that's the problem. Where does the inspiration for a solution like CellFM come from?
Well, what's truly fascinating here is researchers are turning to a maybe surprising source, large language models, LLMs, the tech behind things like ChatGPT.
Right. How does that connect? Language models and cell biology.
Just like LLMs learn the language of human, text grammar, context, relationships between words scientists figured, maybe we can build models to learn the language of cells, how genes relate, how expression patterns define a cell's state.
So like a foundation model, but for biology.
Exactly. Early attempts tried fine-tuning existing LLMs, adapting them, but they often couldn't fully harness the sheer vastness and unique structure of gene expression data. They weren't built for it, fundamentally.
Gotcha. So they weren't quite the right fit, which leads to the big question.
Yeah, what if you build one from the ground up?
Yeah.
Specifically for cellular data, and what if you did it on a scale never seen before?
And that's CellFM.
That is CellFM,
Yeah.
a large-scale foundation model pre-trained on, get this, an astonishing 100 million human cells.
100 million. Wow. How does that compare?
To put that in perspective, it's about twice the size of the largest single-species models out there right now.
Twice the size. Okay.
And it boasts 800 million parameters. Think of parameters as the sort of internal knobs and dials the model uses to learn. That's an 8-fold increase over the previous largest single-species models.
8-fold. So we're talking unprecedented scope, unprecedented power, all designed to unify our understanding of what cells are doing.
That's the goal, a unified framework.
Okay, so before we dive really deep into CellFM itself, maybe let's quickly map out the landscape. You mentioned foundation models for cells. CellFM isn't the only approach, right?
No, definitely not. Researchers have explored a few different strategies, broadly speaking. We can sort of categorize them into 3 main types based on how they handle the gene expression data.
Okay, what's the first type?
First, you've got ordering-based models, things like iSeq, TGPT, Geniformer. These essentially try to predict the rank or the position of genes within a cell's context. It's like focusing on which words are most important in a sentence, their relative order.
Interesting. Focusing on the rank, the order, what's the second type?
Then there are value categorization-based models. Examples are CBERT, SCGPT, UCE. These take the raw gene expression values, which are continuous numbers, and convert them into discrete categories.
Ah, like putting numbers into buckets. Low, medium, high expression, maybe?
Exactly like that. It simplifies the data, which can make it easier for some AI architectures to process, but you inevitably lose some resolution, some detail.
Right, rounding things off, essentially. And the third type...
And that's where CellFM fits in. These are the value projection-based models, like Ski Foundation and Gene Compass as well. Their goal is to preserve the full resolution of the data.
How do they do that?
By directly predicting the raw gene expression values themselves. No bucketing, no ranking. They try to predict the actual precise number. It's like wanting the exact decimal point, not just the rounded integer.
So aiming to keep as much biological information as possible.
Precisely, retaining the maximum fidelity to the original signal.
Okay, so CellFM aims for that high fidelity approach. You mentioned it was trained on 100 million cells. Where did all that data come from? That sounds like a monumental task.
Oh, it absolutely was. Monumental is the right word. The researchers had to gather this data from loads of different public databases, NCVI, GEO, ENA, GSA, import, you name it.
These are big standard repositories, right?
Yes, but the data wasn't neatly packaged. It came in all sorts of formats, raw sequencing files, process tables, specialized data objects used by different software. It was a real mix.
So step one was just wrangling all this disparate data.
Exactly. Before CellFM could even start learning, this massive, messy collection had to be meticulously cleaned and standardized. Imagine collecting books from 1,000 libraries, all in different languages and formats, and then translating and organizing them into one single, coherent library.
That's a great analogy. What did that involve, practically?
Well, a really rigorous process.
Mhm.
They used something called the Synaco-C single-cell database. This involved quality control, like filtering out low-quality cells that didn't have enough genes expressed.
Okay, cleaning up the noise.
Yep. And crucially, standardizing all the gene names. Making sure a gene called, say, gene A in one dataset wasn't called a gene in another. They used the official HGNC guidelines for that.
So everyone's speaking the same language,
Mm.
gene-wise.
Right. Then converting everything into a unified format, a sparse matrix, which is efficient for computation. This whole curation effort resulted in, over 102 million high-quality human cells from almost 20,000 samples.
Wow. And covering different organs, different technologies.
Yes. Diverse data, healthy donors, donors with diseases like viral infections or lung cancer. A really broad snapshot of human biology at the single cell level. This unified dataset is absolutely the bedrock of CellFM's power.
Okay, the data foundation is massive and clean. Let's peek under the hood now. How does CellFM actually learn from this? You mentioned its architecture has 3 core components.
That's right. First up is the embedding module. Its job is basically to take those raw gene expression numbers for each cell and turn them into much richer, high-dimensional embedding features.
Embeddings, like giving each gene a complex digital fingerprint, capturing more than just its activity level.
Exactly. It captures its characteristics and, importantly, how it relates to other genes. This module is smart about handling cells with different numbers of detected genes, too, usually focusing on the most highly expressed ones up to a certain limit, around 48 genes per cell.
So it creates these rich fingerprints. Then what happens?
This is where the core learning happens, during pre-training. CellFM essentially plays this game, a masked prediction task. It randomly hides, or masks, about 20% of a cell's gene expression values.
Okay.
And then it tries to predict those hidden values based only on the genes it can see the unmasked ones.
Ah, like fill in the blanks for biology.
Precisely. It's not just a trick. It forces the model to learn the deep underlying patterns and relationships between genes. It has to understand the context of the cell to make good predictions. That's how it builds this really holistic view.
Very clever. So that's the embedding module and the learning task. What's the next component? You called it the brain.
Yeah, the brain is the ERetNet module. That stands for Enhanced Retentive Network. It's a specially modified version of an architecture called RetNet.
RetNet. How does that relate to, say, the transformer models we hear about in LLMs?
RetNet itself is designed as a highly efficient alternative to transformers. Transformers are powerful, but can be very computationally hungry, especially with huge sequences or data sets. ERetNet takes RetNet and enhances it further for this specific biological task and scale.
Enhanced how?
A couple of key ways. They replaced a standard part with something called a gated bilinear network, which helps training go smoother and improves performance. And they swapped out a standard normalization technique with something called deep norm, which gives much better training stability, especially critical when you have 800 million parameters. Think of it like upgrading the engine parts and suspension for a smoother, more reliable ride with a very heavy load.
Makes sense. Handling that much data needs stability.
Absolutely. And a really crucial part of ERetNet is its gated multi-head attention MHA unit. Now, attention mechanisms are key for models to understand relationships, but traditional ones have a computational cost that grows quadratically with the input size.
Meaning they get slow, fast as you add more genes.
Exactly. They hit a wall. But the attention in ERetNet has linear complexity. This is a huge practical advantage. It means CellFM can efficiently learn dependencies between thousands of genes without the computation blowing up. It's what makes training on 100 million cells actually feasible.
Linear complexity. That sounds like a key enabler for the scale.
Okay, so we have the embedding, the ERetNet brain.
Hmm.
What's the third piece?
The third piece is all about adaptability. The low-rank adaptation module, or LoRA.
LoRA, what does that do?
So you have this massive pre-trained model, CellFM, with its 800 million parameters. Now imagine you want to use it for a new specific task, maybe analyzing a particular type of cancer cell it hasn't seen much of.
Right, you'd need to fine-tune it.
Traditionally, yes. But
Yeah.
fine-tuning all 800 million parameters is incredibly slow and needs a ton of computing power. It's expensive.
Okay.
LoRa's is a technique that avoids that. It cleverly freezes most of the big model and adds just a small number of new trainable parameters, structured in a low-rank way. So you only have to train these few extra parameters. Like adding a small custom adapter to a complex machine instead of rebuilding the whole engine.
That's a perfect analogy. It lets you adapt CellFM for specific downstream tasks much, much faster and cheaper, using far fewer resources, while still leveraging the power of the big pre-trained model. It keeps CellFM agile.
Okay, let's quickly recap this first part. The challenge is that single-cell data is incredibly rich, but also super noisy, sparse, and plagued by those batch effects, making analysis tough.
Right, very difficult with traditional methods sometimes.
And CellFM's solution is this massive new foundation model built from scratch for cells, pre-trained on a staggering 100 million human cells with 800 million parameters. Unprecedented scale.
A real step change in scale, yeah.
The foundation for this was a huge effort in collecting and meticulously standardizing data from all over the place into one unified, high-quality data set.
Can't overstate how important that data curation step was. Garbage in, garbage out, especially at this scale.
And the architecture relies on three key parts: an embedding module turning gene counts into rich features and using that masking game to learn context.
That fill-in-the-blanks puzzle, yeah.
An innovative ERetNet module, the brain, using efficient linear attention to handle the massive scale and learn gene relationships stably.
Crucial for feasibility.
And finally, a LoRA module for agile, cost-effective fine-tuning, letting you adapt the model quickly for new tasks.
Making it practical for researchers to actually use.
All right, so we've dug into the why and the how of CellFM. Now for the exciting part. What can this thing actually do? What does all this power translate into for, understanding biology or medicine? Let's start with predicting what genes actually do.
Yeah, gene function prediction, this is a really big one. We have the human genome sequence, roughly 20,000 protein coding genes. But for a significant chunk of them, we still don't fully know what they do. Their function isn't well annotated.
Like a dictionary with lots of undefined words.
Exactly. And CellFM showed remarkable ability here, particularly using what's called a zero-shot approach.
Zero shot.
Meaning it could predict gene functions accurately without being specifically trained or fine-tuned for that particular prediction task beforehand. It used the general knowledge it gained during pre-training.
Wow. So out of the box, it already has some understanding.
A surprisingly good understanding. For example, they tested it on identifying dosage-sensitive genes. These are genes where having too many or too few copies can cause disease. CellFM outperformed other leading models like UCE and SCGPT by a pretty solid margin, like 5–6% better accuracy and F1 scores.
And you could actually see this difference.
Yeah, when they visualized the gene embeddings using techniques like UAPI, you could clearly see that CellFM was much better at grouping these dosage-sensitive genes together, separating them from others. Its internal representation just captured that property better.
That's impressive for zero shot. What about more complex functions?
It also excelled there. They used gene ontology, or JOGO terms, which describe functions in a more detailed, hierarchical way covering biological processes, cellular locations, molecular activities.
Okay, much more nuanced.
Right. And in these multi-class prediction tasks, CellFM again outperformed models like GeneCompass and UCE, this time by over 1.6% in average AUPR, which is a key metric here. It shows it can handle that complexity and accurately assign multiple functional labels to genes.
So the big takeaway is CellFM can quickly and accurately help fill in those blanks in our biological dictionary, telling us what specific roles genes play, even complex ones.
Yes, potentially accelerating functional annotation significantly.
Okay, so CellFM can accurately predict gene functions, even without specific training for it. For you, the listener, thinking about the zero-shot power, how does that capability fundamentally change the pace or the approach to biological discovery when dealing with the vast, often unannotated world of genes?
Right, beyond just understanding what genes normally do, CellFM can also predict how cells respond to perturbations.
Perturbations, like poking the cell.
Essentially, yeah. How does a cell react if you knock out a gene using CRISPR or maybe treat it with a drug? Predicting these responses is incredibly valuable for things like AI-driven drug discovery. Well, you could use it to identify new potential drug targets, genes that, if targeted, might have a desired effect on disease cells. Or you could explore drug repurposing, seeing if an existing drug might work for a new disease by predicting its effect on relevant cell types. It's like running thousands of experiments in silico first.
Okay, simulating interventions. How well did CellFM do at this?
This is where it got really interesting. They tested it using perturb-seq datasets. These are datasets specifically designed to measure the effects of gene perturbations at the single-cell level. Think datasets like Adamson and Norman, which involve knocking out single genes or even pairs of genes.
And the results.
They combine CellFM's learned gene embeddings, those rich fingerprints, with an existing tool called GEARS, which is already state-of-the-art for perturbation prediction. And the combination, CellFM plus GEARS, consistently outperformed all other single-cell foundation models they tested, and even GEARS on its own.
Better than the state-of-the-art. By how much?
For example, it showed about a 4.75% improvement in Pearson correlation coefficients, measuring how well the predicted changes match the real changes, and a 7% improvement, meaning lower error, in mean squared error compared to GEARS alone. It was better at predicting not just which genes change, but the direction of that change. Does the gene get more or less active?
That 7% lower error sounds significant if you're trying to rely on these predictions. More reliable insights into potential drug effects.
Definitely. Much closer to the actual experimental outcomes.
So, CellFM accurately predicts cellular responses to genetic changes, even outperforming existing tools. For those of you interested in drug development or clinical applications, how could this predictive power specifically speed up finding new therapeutic targets or maybe uncovering new uses for old drugs?
And there's another related capability, almost the flip side. Reverse perturbation prediction.
Reverse perturbation, what does that mean?
Imagine you have cells in a specific disease state, characterized by a certain gene expression pattern. Reverse perturbation prediction aims to identify which genetic interventions, like which CRISPR gene edits, could push the cell back towards a healthy state.
Ah, so finding the potential undo button for a disease process at the genetic level.
Exactly. It's about predicting which perturbations could induce cellular recovery.
That sounds incredibly powerful. Did it work?
They used the Norman dataset again, which has many perturbations, and CellFM showed remarkable success. For instance, it could accurately pinpoint specific combinations of gene knockouts, like targeting CNN1 and ETS2 together, or ETS2 and IGDCC3, as top predictions for achieving a desired recovery cell state.
Top predictions, how accurate were they?
In their tests, CellFM achieved an average of about 81.8% correctly identified recovery perturbations within its top 10 predictions. That significantly outperformed another model, SCGPT, by over 18%.
18% better. That's a huge difference in finding the right combination.
It is. Now, it's currently focused on predicting the effect of gene perturbations, not directly modeling drug molecules yet. But the potential to apply this kind of reverse engineering approach to drug discovery in the future is immense.
Finding that genetic undo button. CellFM can predict genetic interventions to reverse disease states. That really opens up possibilities. What kind of impact could this predictive reverse engineering have on how we approach diseases, perhaps even ones currently considered incurable? What ethical thoughts does that bring up for you?
Okay, moving on from perturbations, another absolute cornerstone of single-cell analysis is just identifying the cells. Cell type annotation.
Right, simply figuring out, is this a T cell, a neuron, a skin cell?
Exactly, which is critical for understanding the cellular makeup of tissues in health and disease. Different cell types do vastly different things.
And CellFM, how does it handle this fundamental task?
It proved highly competent here, too. It generally outperforms several other recent single-cell foundation models, as well as more traditional baseline methods like SVM or SMAP.
Okay, better than standard approaches. What about different scenarios, like within the same experiment versus comparing across experiments?
Good point. In intra-dataset tests, where training and testing data come from the same source, which is easier, CellFM achieved an average accuracy of nearly 93%, beating the next best, Ski Foundation, by over 2%.
Pretty high accuracy.
But more impressively, in interdataset evaluations comparing across different studies, which is much harder due to those batch effects we talked about.
The real-world challenge.
Exactly. CellFM again led the pack, outperforming Ski Foundation by about 2.3%. This shows it's robust, it can handle that experimental variation, and still accurately identify cell types.
That robustness is key for practical use. What about really similar cell types? Can it tell those apart?
Yes, and that was another strength. It showed exceptional performance in distinguishing very subtle subtypes. For instance, telling apart exhausted T cells from activated CD8 plus T cells.
Which is super important in cancer immunology, right? They behave very differently.
Critically important. Exhausted T cells don't fight the tumor effectively. Activated ones do. CellFM, on specific datasets like from basal cell carcinoma, surpassed other models like UCE by an average of 6.5% in identifying these nuanced states.
6.5% better at telling those crucial subtypes apart.
And remember that LoRA module for fine-tuning. They showed that using LoRA significantly sped up the process of adapting CellFM for these annotation tasks, making it much faster without sacrificing performance, efficient and accurate.
Precision, robustness, and efficiency in identifying cell types, even subtle ones. So CellFM precisely identifies cell types, including those subtle but crucial subtypes, even when dealing with messy real-world data. For you, the listener, how might this level of precision change how we understand complex diseases like cancer or autoimmune disorders, where identifying specific cell subtypes is often key to figuring out what's going wrong and how to fix it?
Beyond just identifying individual cells, CellFM can also help us decipher the intricate relationships between genes. Understanding gene regulatory networks, how genes control each other.
Moving from the who to the how they interact.
Exactly. The gene embeddings it learns, and also its internal attention maps, which show what parts of the input the model focuses on, act like a magnifying glass for these cellular conversations.
Can you give an example?
Sure. When they fine-tuned CellFM specifically on immune cells, it preserved known relationships between key immune genes like IL-2, IL-3, and IL-4, which are all involved in the JAK-STAT pathway regulating immune responses.
So it learned biologically relevant connections in the right context.
Yes, and crucially, these specific immune-related relationships were absent when they trained the model only on non-immune cells. This highlights its ability to capture context specificity. It learns what's important in immune cells specifically.
Yeah, context awareness is really important.
Definitely. And if you look at pathway enrichment analysis, basically, seeing which known biological pathways are prominent in the gene relationships, CellFM finds it consistently revealed significantly more enriched pathways compared to traditional methods like just looking at gene co-expression.
Finding more known biology.
More known biology, and it even identified unique immune-relevant pathways that the other methods completely missed. So it seems to be giving a more complete, more sensitive map of the functional interplay between genes.
A more complete picture of how biological processes are orchestrated. CellFM maps these complex gene interactions and pathways, and it does it in a context-specific way. Thinking about therapies, how could understanding these gene conversations better lead to new strategies? Maybe targeting pathways instead of just single genes.
And one final piece ties into that. CellFM can efficiently identify which specific genes are most affected when you perturb another gene.
So tracing the ripple effects.
Pretty much. By analyzing those attention maps, we mentioned seeing which genes the model pays attention to when predicting the effects of perturbing one specific gene. Researchers can pinpoint the likely downstream targets.
How well does that work?
Quite well, actually. For instance, they looked at perturbing a gene called SPI1, which is a master regulator in blood cell development. CellFM generated a list of the top 20 genes predicted to be most influenced by messing with SPI1. Right.
And on average, 18 out of those 20 genes were validated as actual targets of SPI1 in external databases like GP Atlas, which map protein DNA interactions. 18 out of 20 is pretty accurate linkage. Very accurate. And it's allowed them to see distinct pathway activation patterns. Perturbing SPI1 lit up pathways related to things like human T-cell leukemia, virus 1 infection, and acute myeloid leukemia, showing how SPI1 and other genes like CCNB2 and CCNA2 work together in these disease processes.
So it connects the perturbed gene to relevant disease pathways through its downstream effects.
Exactly. Another example, perturbing the gene JUN, which is involved in cell growth. CellFM connected it strongly to the focal adhesion pathway, involving genes like CCND3 and CTNB1. These are crucial for how cells stick together and proliferate processes that go wrong in cancer. CellFM basically provides this detailed roadmap of biological impact when a gene is altered, a roadmap of the consequences. CellFM pinpoints the genes most affected by a perturbation and reveals the impacted pathways. For you listening, how could this targeted insight help us understand things like why some cancers become resistant to drugs, or maybe how to design smarter combination therapies?
So summing up this section, The applications are really versatile.
Yeah, it seems CellFM excels across the board.
Yeah.
Predicting gene function, even zero shot.
Predicting perturbation outcomes both forward and reverse.
Precisely annotating cell types, handling batch effects, distinguishing subtypes.
And deciphering those intricate gene relationships and pathways.
Which translates into potentially actionable insights for AI-driven drug discovery, maybe more personalized medicine strategies down the line.
And just a fundamentally deeper understanding of complex biology and disease mechanisms.
So what is pretty big here? CellFM isn't just another research tool. It feels like a new kind of window into the basic operating system of our cells.
Yeah, it really promises to accelerate biological discovery and hopefully therapeutic development.
Wow, what an incredible deep dive into the world of CellFM. We've really seen how this state-of-the-art foundation model, trained on that unprecedented scale, 100 million human cells with its smart ERetNet architecture and 800 million parameters, is truly pushing the boundaries in single-cell analysis.
It really is. From accurately predicting what genes do, forecasting how cells will react if you poke them, annotating cell types with high precision even across different experiments, and mapping out those complex gene networks, it consistently seems to outperform existing models in benchmark tests.
That ability to just extract meaningful insights from these huge, messy data sets feels genuinely transformative for biology and medicine.
It really does.
It's such a powerful demonstration of how large-scale AI can maybe give us that shortcut to being truly well-informed about the fundamental processes of life. Now, it's a massive leap, but the researchers themselves acknowledge limitations, right?
Oh, absolutely. Science always has next steps. They mentioned the current focus is on human data, so single species. And they acknowledge that fully interpreting the why behind its predictions, especially from those attention maps, still needs new explainability techniques to be developed.
So understanding how it knows what it knows is still an area of research.
Yes, but the future looks bright. They have plans to expand its capabilities, for instance, to handle drug perturbation datasets directly, not just genetic ones. That would be a huge next step for drug discovery applications.
Definitely.
And importantly, in the spirit of research collaboration, they've made the CellFM code and the pre-trained model publicly available.
That's great open science.
Yeah, it means researchers anywhere can download it, use it, build on it, leverage this unified framework to push their own specific research forward. It's designed to accelerate discovery for everyone.
So what does this all ultimately mean for you, the listener? CellFM and models like it offer this potential shortcut to understanding the incredibly complex language of our cells. It's like getting a peek behind the curtain of biological function and disease.
Imagine the possibilities when we can truly understand and maybe even predict the language of every single cell in our bodies, in health and across diseases like cancer or Alzheimer's.
The potential feels enormous. So, a final question for you to ponder. What other biological mysteries, what big unanswered questions in life sciences do you think models like CellFM could help us unravel next?
Good question to leave folks with.
Thank you so much for joining us on this deep dive today. We really hope you've gained a clearer picture of this groundbreaking work and that you're as excited as we are about the future of AI and unlocking the secrets of life itself.